introduce themselves, talk a little
Cool.
bit about your research.
All right. Well, I'm a developmental biologist, which means I study how you go from being an egg to being you with all the parts in the right place. What draws my attention to biology really are the patterns that exist and how things fit together. At St. Mary's College of Maryland, I'm a professor in biology. And probably one of the most important things we do is to mentor and direct 6 or more research students per year in our labs, doing actual wet lab science. Most recently, probably one of my most important manuscripts has been the application of CRISPR-Cas9 genome editing in cephalopods. And I was the first person to demonstrate proof of concept that this was possible. And so with my research at St. Mary's, we study axolotl limb regeneration. We study worm regeneration in a freshwater worm called Lumbriculus variegatus. And we study squid embryos that I bring back from Woods Hole. And so some of my students have their hands in a variety of systems. But I'm mostly just a faculty member mentoring a lot of students, helping them find their way and their direction in life.
So basically, I have 2 questions that I want to guide the rest of this conversation. We'll dive into some of the deeper science. And the first one is just about humans and how they heal. And then sometimes it seems like when you exercise or you stress your body, it heals back stronger. And that's why we train. We train to be stronger. But sometimes we stress it too much and we hurt ourselves. And I want to know kind of where that line is. Yeah. So I just want to compare those 2 processes, if they're different at all. Maybe there's some sort of spectrum and we can try and understand that. And then to go even further down it, you study regenerations, but that's not that you're damaging and healing it now. Now you're either destroying it or removing some of this organ tissue, and some animals can just regrow it. And so that's again, maybe it's a spectrum of between the healing of the damaged thing and regeneration, but I want to understand basically this whole spectrum and how it works, why we do these things, why animals evolve these different abilities. And yeah, we're just gonna try and understand this whole thing.
All right. The whole notion of healing and why some animals heal really well and some animals don't. And I think healing is a compromise. Healing for us as a mammal is frequently counterbalanced with our desire not to have an infection and not to be infected and to die. So I think part of the reason we heal and scar the way we do, for example, our skin, is because we want to close that wound down as fast as possible, and we want to protect ourselves, the insides of ourselves, from infection. Healing also is impacted by age. So young children are going to scar differently, and certainly neonatal children in the womb don't scar. They don't scar until after they're born. Axolotls don't scar. That's the Mexican salamander. And there's a spiny mouse in Africa that doesn't scar either. So that's a mammal. And folks are really looking at like, okay, why does this spiny mouse not scar when regular mice and humans do? So when you exercise and you put stressors on your muscles, what you're doing is you're actually making really tiny little micro tears in the subcellular structure, the actin and myosin filaments that slide by each other that allow your muscles to contract. And you're making micro tears in that structure. Your body is constantly replacing actin and myosin proteins. And so with those micro tears, you build them back stronger, you build them back so that they're more capable of withstanding the stressors that you gave them. This is why when folks lift, they lift arms one day, legs the next day, arms one day, legs the next day. You have to have a moment in time where your body can recover from those small stressors. So muscles can become stronger. Bone responds to stress against it when you exercise, when you use your body. And that's because bone is laid down by bone cells called osteoblasts. They lay down new bone, and it's chewed up by osteoclasts. And And the reason we have osteoblasts and osteoclasts, and osteocytes are just, card carrying, I'm a bone cell, I'm doing my job. The reason we have osteoblasts laying down new bone and osteoclasts chewing up existing bone is because it's really our calcium and our phosphate bank. And calcium and phosphate's really important for nucleic acids and for ATP and for neural function and muscle function. And so the calcium and phosphate that we ingest is deposited in the bank. It's deposited by the osteoblasts and it's freed up by the osteoclasts to maintain homeostasis of blood calcium levels and phosphate levels in your blood. When you're a little kid, and certainly your beautiful daughter she's growing like crazy. So her osteoblasts are winning and her osteoclasts are not really doing much at all. So she's really growing fast. Hit adolescence, young adulthood, and it's really a balance. Osteoblasts, osteoclasts doing the same thing. You get to my age, you pass menopause, and all of a sudden the osteoclasts start winning and the osteoblasts are lazy. And some of the treatments for menopause actually stimulates the osteoblasts and quiets the osteoclasts so that your bones can stay stronger. When you exercise and you put stressor on that bone, so weightlifting, weight-bearing exercise, that stimulates the osteoblasts to lay down more bone and to make your bones stronger. Now we talked about hurting yourself. So that's the bony part and the muscle part. What connects all these things together are ligaments and tendons. Ligaments are going to hold the bony elements together, and tendons are going to hold the muscles onto the bones. And there's a variety of different ways that they do this. But what most folks probably aren't aware of is that ligaments and tendons don't have a direct blood supply. They're avascular. And so in order for a ligament and tendon to become stronger, they have to actually diffuse nutrients and diffuse wastes in and out of this non-vascularized tissue. That takes time. If you're an athlete and you tear or you overstretch that ligament or that tendon, this is why you have to rest 4, 6, 8 weeks, depending on how long it's been since you've injured that element, because it takes time for the waste products from the tear to get out and the good stuff to get in, and you can't rush it. Heat can facilitate vascular flow around a joint and it may feel good, but cold may actually slow down the degradation process of the waste products. And so I'm a big fan of cold, especially when it comes to a spasm or to trauma to a joint, because that helps to reduce the immediate inflammation. It also works to break a muscle spasm, because nerves actually grow through muscle fibers. And if the muscle spasms and it pinches on the nerves, they're going to release more neurotransmitter, which causes the muscles to contract more. By chilling everything down, everything relaxes and the nerves can quiet. And it's really as simple as chemical reactions go faster when it's hot and slower when it's cold. And so to really get that, so if someone has a pulled muscle in their lower back, sitting on a bag of frozen peas with that right behind their back for a little while can allow those muscles to relax and stretch out. And then you just have to be mindful to sort of slowly bring it back to some kind of reality.
Yeah, good. So I really just wanted to start, I don't have much experience with biology. So start from the beginning of cells dividing and why we have this churn of cells dying and reproducing in our body, where the dead ones go, where the new ones come from, and just why do we have this process of just churning through cells in our body? What does that do for us? Yeah.
Sure. All right. Well, so one way to think about this is we're constantly renewing our tissues. There are some cells we don't renew at all. We replace parts of them, but we don't renew them. And your muscle cells are largely the muscle cells you were born with, which is always kind of crazy. Like your beautiful little daughter has her muscle cells. and they're going to grow, they're going to elongate. She's going to add fibers to them. Some of the bodybuilders can force their muscle cells to split and get more cells that way. But they're not a cell population that really replaces itself. It grows and then it replaces the parts inside. The cells that are most actively growing and dividing on us are skin cells that we slough off from the outside, but also we slough off from the inside of the digestive tract, and our blood cells. The cells that I think are most actively dividing are the blood cells and the epithelial cells that line our digestive tract and cover our bodies. I want you to think about your body like a donut. So if I could take myself and shrink myself down into a tiny little person and hold on to a string, and that person went into my mouth and down my esophagus and into my stomach and through my duodenum and my jejunum and my ileum, my ascending, transverse, descending sigmoid colon. That little person would walk all the way through me through a tunnel, because what you eat, what you ingest, the food that you take in is never really in your body. It's in a tube that passes through your body. And that tube is lined by, in most places, a 1-cell layer thick layer of cells. It's not multiple layers of cells. So what's protecting you from the tortilla chips that you ate is 1 layer of cells. And then a basement membrane. Interestingly, that layer of cells doesn't have its own blood supply either. It's got to get everything by diffusion. So the cells that line your digestive tract live for about 3 to 4 days. That's it. Then they divide and they leave one behind and they die. One dies. And so you're constantly sloughing your own cells into your digestive tract where you eat them, you digest them. And you recycle the nucleotides and the amino acids that make up the cells. Your skin is constantly replacing itself, but this stuff on the outside is kind of dead. So it's waterproof and bug proof, and bacteria can't get through it unless you cut yourself, which is why we use Band-Aids and cover our wounds. The cells that make our hair and our nails are also epidermal cells that form this structure. So if someone, so we have actively growing skin cells, actively growing hair cells. And when you think about someone who has cancer, they have an actively growing population of cells, and we want to quiet that down. We want to get rid of it. And so there are chemicals that interrupt the cell's ability to divide. They inhibit the cytoskeletal, the spindle mechanism that allows chromosomes to separate and cells to divide. And so when someone's on chemotherapy, what happens? Their hair falls out because the follicle cells become quiescent. They have nausea because the digestive tract is not being replaced. And they have anemia because they're not making the millions of red blood cells and thousands of white blood cells that they need every day to replenish those cells in the body. And this is why they become anemic and nauseous and their hair falls out. But we've come up with ways to make those symptoms more tolerable, but those are the actively dividing cells in our body that are being replaced all the time. As we get older, things slow down. So I don't make as many red blood cells every day as I did when I was your age. My skin becomes thinner and more easily wounded and bruised because it's not as thick and plentiful. And for some folks, their hair thins and their hair falls out. And so, well, there's other reasons why hair falls out, but there are certain times in our lives where we are really robust and churning and everything's working great. And we are not really susceptible to illnesses because our immune system is fighting fit. But as we get older, things change, things slow down, we become more vulnerable and we don't repair as quickly.
So you talked mostly about or tissues that seem to grow in a direction. Your skin kind of sloughs off, your digestive tract grows in and sloughs off into your digestive tract. And then we did talk a little bit about bones and muscles that they don't really have a direction to go. I guess they go to the outside, but it's still in your body. And I'm just wondering where how those kind of growth mechanisms and replacement mechanisms work when there's not an obvious direction like there is to some of those other tissue.
So that's a really fundamental question. What makes the structural stuff of our cells are proteins. Okay, and proteins are built from amino acids, and amino acids are coded from messenger RNAs that are produced, that are synthesized or copied, transcribed in the nucleus, and then they go out of the nucleus into the cytoplasm, and that message engages ribosomes, these protein synthesis machinery mechanisms, and other nucleic acids called transfer RNAs come in with amino acids, and you make more proteins. So when my muscle cells want to make new actin, my nuclei transcribe mRNA for actin. The mRNA goes out of the cytoplasm, amino acids line up, and I make a new actin globular monomer. Then they all line up like beads on a string, and I can make an actin filament. So muscle is, I want you to think about muscle as sort of just constantly recycling out old fibers and making new fibers, but it's still the same cell. And it's also still the same nuclei that are in that cell. Bone recycles itself. It's got a very rich blood supply. It provides nutrients. Osteoblasts lay down new bone. Osteoclasts chew bone up. And the osteocytes, they have a finite period of life, but then they're replaced by new osteoblasts, which differentiate into bone cells. Bone is responsive to stress. And so the easiest way to think about it is many, many people have experience with oral braces, wearing orthodontics. And what oral braces do is they actually put brackets on your teeth, and they exert stress against your teeth to drag your teeth through the bone of your jaw. Imagine a carrot in the ground, and the root of your tooth is the carrot, and you're grabbing the top, and you're just like pulling it, and you're trying to drag that carrot through the ground. And so after someone has orthodonture, frequently the roots don't necessarily go like this. They can go like that. So the tooth's over here now, but the root remembers it's over there. And this is what retainers are all about. But what allows orthodontics to work is the fact that you're putting pressure on one side of the tooth, and the osteoclasts break down the bone, and there's less pressure on this side, and the osteoblasts fill it in. So because of the pressure, you're actually dragging that tooth through the bone, and the osteoblasts and osteoclasts are letting you do that. Now, other cells that don't divide in the body, neurons, for the most part, don't divide. Now, there are a few interesting places where neurons can divide, and we're learning more about that. For example, birds acquire new songs every year, and it seems that that's associated with new neurons dividing. For the most part, we're born with the neurons we've got. Your daughter is born with all her egg cells. She's got them all, and they're dropping dead like flies right now, and we don't really understand why. We don't understand why when we're born, we've got all these beautiful egg cells, and then a whole bunch of them just poop out. In the male species, sperm divide like crazy. Spermatocytes divide like crazy, and we make sperm all throughout life. And so there's differences in different tissues, different body parts. Your liver is unique in that, and this might be why it's such a good system for regeneration. Liver cells are hepatocytes. They are doing the liver cell job, and then they can divide while they're doing that job and make two liver cells that are doing that job. Normally, when cells divide, they look more embryonic-like, and they're not doing that job. You can give 60% of your liver away, and your body will respond, and your liver that you have left will grow and restore the physiological function of the original liver.
And that's actually a good segue to my next question about, so your liver grows to a certain size and the
Yep.
cells are kind of organized in a way where they have a place and they know where they're supposed to be and they don't get bigger than that. And so somehow they're communicating to each other to fit this structure and a certain size. And I guess you can manipulate it in some ways, like you talked about with the orthodontics, you can move things around, but there's still a trail and they still kind of know where things are and where they
Yep.
want to be.
Yep.
So can you explain like how the cells kind of signal to each other and know where they're supposed to be?
That's a good, that's a $6 million question, isn't it? One thing we know is that cells talk to each other by their cell surfaces. So if we take cells from a chicken embryo and we disassociate those cells and we put them in a petri dish, those cells will divide and they'll create a one layer confluent layer of cells. And once everybody is touching the edges and each other, they stop dividing. And we really don't know why. You can take muscle cells and nerve cells, put them in a petri dish, and the muscle cells will go this way and the nerve cells will go that way, especially if you exert an electrical current across the field. It seems to enhance the likelihood of a neuron hooking into a muscle cell. For example, why do the lungs stop growing? And then with physiology, why and growth of the individual, why do those lobes enlarge as we grow and reach adulthood? I don't think folks really know. We do know that with stress, the heart can enlarge. With exercise, the heart can enlarge. And those are very, very different things. So in response to demand, an organ may become larger. I've heard that if you give one kidney away, the other one will grow a little bit to compensate. So there is this feedback of physiological need, homeostasis, what does my body need, and what is it missing, and the development of cells. A good example is giving a pint of blood. So you go and you give a pint of blood. And your body's like, wait a second, I need a little bit more oxygen up in this brain. So the hearts may beat a little faster for a little bit. You may have to take in more nutrients. And the reserve blood cells in your bone marrow, your spleen, and your liver are all going to go like out into the body system. So you've used your reserve, and what stimulates you to make more blood cells is the need, the homeostatic need. Some folks have a disease called polycythemia, and that's a cancer of the blood, the red blood cells. And so polycythemia, they make too many red blood cells. Those individuals actually have to go to the doctors and be bled so that so their blood isn't so thick that it's not difficult to pump and they don't clot unnecessarily. And so it's this, what regulates growth, what regulates enlargement, what regulates the creation of more red blood cells. It's all this homeostatic negative feedback loop of, I need a little bit more oxygen, I'm going to make a few more red blood cells. This happens when folks fly into Denver. All right, so they fly into Denver and they go to the Mile High City, and all of a sudden the air is much thinner. And so for the first 2 or 3 days, they may have heart palpitations. They may have trouble falling asleep at night. Their heart may race. They may get winded walking up a set of stairs that they'd never have trouble walking up in Illinois or Massachusetts. And then after 3 or 4 days, everything's pretty much okay. That's because their spleen and their bone marrow and their liver have basically shot out every reserve red blood cell they had, and their body's starting to make more. And this involves communication between the kidney and the bloodstream. The kidney senses oxygen levels. I know that seems kind of weird because you think about the lungs and the heart and the brain thinking about oxygen. But the kidney senses oxygen levels, and if the oxygen levels are not high enough, it releases a chemical called erythropoietin into the bloodstream, And that jazzes up all the stem cells to make more red blood cells. And it takes about 3 or 4 days. This is also why athletes sometimes visit or exercise in hyperbaric pressure chambers where they can be exposed to low levels of oxygen to stimulate synthesis of red blood cells.
What I understand from you telling me then, it's not this like top-down approach command. I don't know if your brain would be saying, it's not your brain telling your body to react in this way. It's your cells that are kind of reacting to stimulus from, lack of oxygen, stress here, whatever's going on. Your individual cells kind of have the instructions and act almost autonomously then.
Well,
Is
all
that
right.
right?
So there's a whole nother nervous system we're not talking about. All right, so we've got the brain controlling the show. But what really controls this homeostasis is something called the autonomic nervous system. And there are 2 branches of the autonomic nervous system, the sympathetic nervous system and the parasympathetic nervous system. And so these things happen without your brain being involved. So the sympathetic nervous system is sometimes referred to as the fight or flight system. So you're driving your car down the highway and there's a semi in front of you and you decide, I'm going to go around this thing, and you go to the left and there's another semi coming right at you and you have this adrenaline rush and you bring your car back behind the semi and you don't die, which is good. Fight or flight, adrenaline, noradrenaline, release of adrenaline from the adrenal glands on top of your kidneys. The parasympathetic nervous system is often referred to as the rest and digest nervous system. That's the one that helps you digest your food, reduces your heart rate, respiration rate. So sympathetic's going to start that heart rate going, increase your respiration rate, get like oxygen, nutrients, things moving fast into your bloodstream. And so it's really the mix of sympathetic and parasympathetic innervation, as well as the hormones, adrenaline in this case, or other hormones that say, okay, it's time to sleep, it's time to rest, it's time to digest, it's time to run away from the bear that's going to try and kill you. And so the cells in your body are responsive to the signals from these 2 elements of the nervous system, as well as to the hormones that are circulating in your blood. Say a woman becomes pregnant and she starts to produce the hormones of pregnancy. One of the very first indicators that there's a change, besides the lack of menstrual flow, is that her breasts become tender and they grow. And that's all hormonal. And the breast tissues enlarge and they differentiate. And in 6, 7, 8 months, they're starting to produce milk. And once the baby is weaned, there's no call for that milk. The tissues resorb, they go back to their quiescent state. And that's another level of homeostasis. So once you've produced milk and you've fed a kid, you don't do it for the rest of your life. So the body tissues can cycle in some cases through periods of activity and periods of quiescence. This is also why I think they were so lucky in getting the first mammalian clone, which is Dolly the sheep. And Dolly the sheep was named Dolly because they actually took a mammary cell to clone into an enucleated egg to make Dolly. And I often wonder, I'm sure they tried every other cell in the body they could get their hands on, but the fact that memory tissue cycles into activity and then quiescence and can be reactivated with another pregnancy over and over and over again probably demonstrates some sort of DNA plasticity that allowed that to happen.
Basically, you're describing your cells respond mostly to chemical signals
In
and contact with each other.
And contact with each other. And so they're basically sensing their environment all the time. Yeah. They're getting chemical signals. They're in need of oxygen and nutrients and wastes. They're connected to their neighbors. If they're in epithelia, they're very tightly connected. And so they're aware, they're sensing their environment all the time.
and they have some sort of internal instructions that know how to react to its environment then.
Seems, I mean, I never really thought about it that way, but I guess I just accept the fact that my heart muscle cells know to contract with their other neighbor heart muscle cells and repair. So with repair, so for example, if I think about the respiratory epithelium that lines your larynx, your voice box, and your trachea. If you over traumatize the respiratory cilia in the respiratory tract with smoking or heavy chemicals in the air, the body responds to that by changing what the cells look like and making them more skin-like, making them stratified and squamous-like. So they don't move the mucus because they don't have cilia, and you get these patches of multiple layered cells that are trying to protect your respiratory tract in another way, but they don't function the same way, which is not a good thing. It's your body's last effort to try and protect you. Just as if you play an instrument or you rake in the garden and you use your hands a lot, you develop calluses in athletics or whatever. Your skin will actually get thicker and heavier in order to protect you. In some ways, you can imagine the lining of the trachea, the tube that goes to your bronchi and your lungs, gets so irritated that it actually thickens and becomes a flatter squamous epithelium like your skin.
Yeah, so with those calluses, then they have some time delay to respond to basically being stressed out, and somehow they know it's not like your brain telling you, okay, we need to be stronger here. It's them responding to something.
They're
And then
responding to mechanical stimulation. Yep.
And then as you stop that, they'll slowly disappear and get back to this homeostasis.
Although I think for some individuals, I can tell when I take bloods for my students, when we test their blood types and such, even though I'm wearing gloves, when I touch their hands, I know if they do something stressful with their digits because of just the feeling of their skin. And I wonder if once you've really used your hands heavily for your whole life, if they ever really go back to being sort of super baby, thin, soft. I don't know.
Your body's always balancing too, I guess. It's not optimized for everything. Of course, it can't optimize for everything. So there's trade-offs for all these things. Your body's not just like, we're going to be awesome at everything. Often, I guess it's nutrition, calorie, or nutrient balance, but there's probably other things where you're balancing functionality of different organs against each other, and that's probably important for healing as well. And just
a good portion of this is regulated by negative feedback loops, so we don't sort of spiral out of control. I think about heart rate, blood pressure. If your heart's beating too hard, there are sensors in the aortic arch that are like, well, hey, this is too much. And that actually allows the parasympathetic nervous system to quiet the heart. And so these feedback loops are there to try and keep everything in a sort of an even playing field. There are very few positive feedback loops, one being birth. So basically, woman's in labor, the muscles contract. They don't relax all the way out. They contract, then there's a pause, and then they contract, and then there's a pause, and then they contract again. So they contract until they get that baby out. But there are very few positive feedback loops because things would just spiral out of control.
We talked a little bit about cuts, but you want to go through
Sure.
just that healing process, and because we talked about signaling, and there's all these different stages, right? And your body kind of knows how to go through these healing process.
So let's just talk about your skin and a cut. Yeah. And also let's talk about rug burns, because when you have a rug burn, you're not bleeding, but it still hurts. That's because nerves actually grow up in between the outer epidermal cells. And so you have nerve endings in your epidermal cells, and you actually have immune cells that crawl around in your epidermal cells. But if you manage to compromise the epidermis through something called the basement membrane, which is the acellular mat that everything's got to diffuse through from the blood vessels down here in the dermis. And if you've ever had a good cut, maybe you saw sort of, it looked a little white, there's blood in this, that's the basement membrane. That's the collagen between the outer cells that are avascular and you, where all the blood vessels are, the dermis. So the first thing that's going to happen is pain. And the pain receptors and blood is going to cause the blood vessels right there at the cut to close down. So they close down really tight. And the clotting factors in your blood are exposed to the collagen fibers on the outside of your blood vessel. And that stimulates them to start to clot. So they make this little mesh of cells. And the smooth muscle in your capillaries closes down to prevent bleeding too much. This is like with a small cut. And you form this hematoma, or this little blood clot. The next thing that happens is the capillaries that are near this, they actually send out chemical signals that tell the white blood cells, hey, there's a compromise here, get over here, you've got to clean this up. And the white blood cells come and invade the clot and start to gobble up any bacteria or dead cells or things that are there. It also stimulates the cells, the epithelial cells on each side, to proliferate and sort of try to grow underneath this clot. This is why if you've ever picked a scab, it's sort of mostly smooth underneath because the cells have already gone underneath that scab. And if you just sort of let it be, it'll pop off by itself. And so they've proliferated, gone underneath, and then the epithelial cells, the basement membrane's restored, and the epithelial cells restore the layers. What happens in the dermis, the dermis, if you cut all the way down through the dermis, you've messed up the collagen fibers that are there. And so cells called fibroblasts come in and they lay down collagen as best as they can, willy-nilly, to repair that dermis. And they don't do it perfectly, and that's why you end up with a scar. And that scar will never be as supple and as movable as the skin was in the beginning. The collagen fibers actually run in your body in preferred or preferenced ways. So now I want you to take your fingers, and I want you to pinch your upper arm, and you can bring the skin in. Goes in easy. Turn it 90 degrees and try and make it go the other way. That's because you've got lots of collagen fibers running this way, but not so many running that way. This is why hand surgeons and face surgeons get paid so much money, because there are collagen fibers running in patterns in these tissues. So when they're compromised, they know how to sew up that wound just the right way so that when you smile, hopefully you won't have a little pinch. Another place where you can visualize without a cut the collagen fibers under supporting the dermis to the basement membrane to the epithelium is in the formation of stretch marks. So if someone gains a lot of weight or grows very quickly, this happens in men as well as women. Pregnancy for some women who have different collagen compositions in their abdominal and breast skin, because it's growing so quickly, the connective tissue, the collagen in the dermis can stretch and tear, and it leaves what are called stria or stretch marks. That never comes back. Once those collagen fibers are torn, they can't be rewoven, and the skin is different than it was before the growth or the pregnancy. Now, it's a genetics thing. Some folks have very pliable collagen, some folks don't. And so it's whether or not you're going to get stretch marks is more of a factor that relates to whether your parents did and the kind of collagen fibers you've got. So sometimes it can take a wound, a cut may heal in a week or two, but the remodeling of the connective tissue and the creation of the scar tissue underneath could take weeks to months.
Yeah. So we talked about your cells' normal function, how they're just kind of multiplying or dividing and, trying to fit together, react a little bit to their environment, get stronger, thicker, whatever. And then it's the same cells. I guess they bring in some outside cells to help, but basically when there's this emergency, they also have these emergency instructions where they just like, okay, now we're in emergency mode and we're doing something different now.
Well, with the emergency mode really just the two important changes are the initial constriction of the blood vessels to prevent blood loss, the release of chemicals to signal the white blood cells to come to clean up the mess. So a bruise, this is good. So you say you get a hematoma or a bruise underneath your skin and you can see it. And it's usually initially very dark, bluish brown. And then it gets more greeny yellow, and then yellow at the end. And then all of a sudden it looks more normal, but it takes a long time to get those, to go through those changes. What you're actually visualizing with the different colors of a bruise are the white blood cells eating the wasted hemoglobin from the red blood cells that have basically died and created that hematoma. And if you want an example of watching your skin cells grow, if you've had a blood blister, it sort of makes this perfect little round dot, maybe often on your hands or place where it got pinched. It's this perfect little round dot because basically the blood vessels underneath the basement membrane have bled into and created like a little space in between your epidermal cells. And then as the epidermal cells proliferate, the little dot just rides all the way to the surface and then is shed when it gets to the top. So what a blister is the epidermal cells and the dermis sort of shifting by each other and at the basement membrane, and it creates a fluid-filled space. And that fluid-filled space is actually between the dermis, where all the blood vessels are, and the epidermis, which is all the cells that are dividing and then eventually dead on the outside. So a blister is because the cells aren't held together perfectly. Now on our fingertips, the epidermis and the dermis go up and down, they interdigitate. So it's a lot harder to get a blister on the end of your finger. Not here. We don't have that same interdigitation here. It's more like this, and you shear when you rake and you get a fluid-filled pocket, and then you get the blister.
Yeah, I always noticed that where I used to pop the blisters and then I noticed they heal faster if you just let them go, right?
Yep.
And yeah, so is that because at least in part you're saying how basically you balance healing with infection, right? And you also want to be able to get nutrients and waste in and waste out, so you want it to be permeable at least to some things. How does that work for a cut? Because it scabs over, it puts this hematoma on that kind of is meant to protect, and
so it scabs over, and the collagen fibers start to try, if it goes deep, it takes a while to lay down that collagen, but the epithelial cells on the sides that are still okay, they grow in and they basically undercut this hematoma, and then they proliferate and come up, and that's why the scab comes up and lifts off.
I see. Okay, so it's not replacing them inside the hematoma, it's growing underneath and pushing it up.
Exactly. Yeah.
Interesting.
The scab, the hematoma, if you have a cut and you have blood, that's going to form the scab. And the scab is actually going to contract. All this dead stuff is going to contract. And sometimes as it's contracting, it releases fluid. And that straw-colored fluid is basically plasma. So it's basically the same thing. It's your blood plasma without clotting factors, because the clotting factors are all in the scab. And then the whole thing is just lifted off. So what your body's trying to do is trying to put a very fast hematoma scab band-aid on the wound and then grow underneath to protect you, so that we don't want bacteria to get into your bloodstream and enter in that way. So that's why it's good not to pick your blisters, because you're creating a direct pathway into your bloodstream for any kind of infection.
I see.
But our bodies are pretty good at fighting most infections, except for when things like pandemics happened.
Okay.
And there are a lot of white blood cells constantly crawling around in your lung tissues looking for problems, looking for bacteria. And you have cells actually in your skin that are crawling around looking for bacteria. You have an incredible surveillance team. There are white blood cells in your brain tissue called microglia that do surveillance on your neural cells every day. And they're always looking for problems. And if they see one, they take it out. So you have this wonderful immune system that's constantly doing surveillance on your body, looking for things that don't belong, or cells that have died, or cells that have are abnormal, like in cancer cells. We all get cancer cells, but our immune system usually takes them out before we recognize them.
We talk a little bit about bones too. At least the injury seems to have a similar shape. Maybe it doesn't have the same layers of the skin. It's not trying to protect against an infection from the outside necessarily, but it's still a damaged part of the tissue.
So a bone, so you can bruise a bone, and actually you've got a very tight membrane on the outside of your bone called the periosteum, and it's what the muscles invest into. And you can bruise that, and if you get a hematoma underneath that, it hurts a lot. It takes a long time for the white blood cells to get in there and chew that up. If you break a bone, you can have different kinds of breaks. You can have a non-displaced break, so it's just a hairline fracture or a twist up the bone, and the first thing that happens is a hematoma. Believe it or not, it doesn't seem like it could do much, but the first thing that happens when you break a bone, because it's so vascularized, is you get a blood clot. And cells come in and lay down connective tissue. And the next thing is you form what's called a soft callus, which is made from a type of cartilage called fibrocartilage. So you have this blood clot, hematoma, that's replaced by granular tissue, then replaced by cartilage, and then replaced by bone. And so, but it's the same thing. You'll have a blood clot, white blood cells come in, that's the granular phase. Then fibrocartilage cells come in to try and make a little stronger splint between those broken bones. Meanwhile, osteoclasts are chewing up the dead stuff, and osteoblasts are trying to lay down the new stuff. And then you get a hard callus, and over time, everything remodels back, so you get your bone marrow cavity back. But you will be left with a scar on the bone. So when you break a bone, bones heal stronger at the break than they were before the break. And I have a skeleton in my anatomy lab that has a very common break. It's an ankle bone. It's a fibula ankle bone break, and it healed beautifully. It was probably non-displaced. The only way you can detect that the bone was ever broken is to actually run your hands down both sides of the skeleton and feel it. So I always challenge the students to find the break. So bone tissue is very well vascularized. There's a lot of blood cells in it. You've got marrow cavity in many places. There's an outer membrane, the periosteum, the inner marrow cavities, endosteum. Cartilage is a whole nother bag of beans. Cartilage is not vascularized at all. I want you to think about, say you've got 25 people in a room, and they're all standing in the room, but they can't touch each other. And you fill the room halfway up with Jell-O. Cartilage is kind of like that, cells sitting in matrix in a room. So you can't touch each other. And so the only way you get nutrients to the cartilage cells is if it diffuses all the way through the Jell-O. The only way you get rid of your wastes is if you diffuse all the way out through the Jell-O. And that's why cartilage and tendons and ligaments heal so slowly, because good stuff's got to diffuse in and the waste stuff has to diffuse out. Bone, these cells sit in little cocoon spaces called lacunae, and they send little processes out to all the other bone cells. So if there's a center blood vessel and then rings of bone, the bone cells are between these concentric rings, and they send out cell processes to their neighbors. So from the blood in the middle, this bone cell, this bone cell, this bone cell, this bone cell, it's like a bucket brigade. They pass the nutrients from one to the next, and they pass the wastes back too. So you're kind of stuck passing nutrients and wastes to your partners in order to allow bone to have its morphology and its shape. But bone has a lot of nerves. That's why it hurts so much when you bang your shin up against a table, or when you break a bone, and it has a lot of blood vessels.
So it has actual vessels through it. It's filled with tunnels.
It's filled with tunnels. They call it the Haversian canal system. And then every once in a while, and most of them run the long length of the bone, but then they'll have some side ones called Volkmann's canals that go sideways. And so all of your bone cells are constantly being exposed to nutrients, oxygen, waste disposal, and the like. But the cartilage cells, and you have 3 different kinds of cartilage cells. So if you feel the tip of your nose, that cartilage is hyaline cartilage. Hyaline cartilage is also the cartilage that's at the end of long bones. So if you have a chicken wing, and you'll see that nice white, shiny, almost looks like milk glass. It's perfectly white. That's cartilage. So the next time you have a chicken wing or a chicken leg, take a knife or a razor blade and just slice through the cartilage. It cuts kind of like water chestnuts. It's very satisfying. And if it's fresh, if you haven't cooked it, if it's fresh chicken and a fresh joint, as you cut through it, you'll actually start cutting through the bone. You'll cut through the compact bone on the outside. You'll cut into the bone marrow cavity. You'll be able to see it all. And you'll say, wait a minute, I'm cutting through bone. Bones are hard. Bones aren't hard. Dead bones that have no organic molecules in them, they're hard, they're brittle. But our bones are actually pretty flexible. Your daughter's bones are like rubber, which is a great thing, This is also why children, if they do fracture their bones, often they refer to them as green stick fractures, because they've got so much collagen and so much of the springy organic stuff that they don't snap. They sort of more fragment, like trying to break a green stick to roast a marshmallow at the fire. Our bones, as we age, the organic components of our bones, the collagen and connected extracellular matrix proteins, they become less prominent, and the bony brittle stuff becomes more prominent.
Okay. So when we get a scar though, why are scars at least seemingly permanent then? Why doesn't our body eventually sort of replace that with the normal tissue of the skin and bone around it?
And that's a great question. And this is a question that folks are looking at. They're looking at these axolotls that I study, because axolotls don't scar. You know, I can make a cut in them, they heal, they form beautifully, they don't scar. And this African spiny mouse doesn't scar. And there are probably a lot of organisms out there we just don't know about, and that don't scar very much. But so we're looking at why our fibroblasts and white blood cells come in and lay down scar tissue, as opposed to just healing perfectly as if it was developing. And my guess is it's just the trade-off of trying to repair so you can go on with function. As it's repairing, you're continuing to move it and shift it around. And so there's probably stressors on that. But basically, once we're born, we scar. Little kids don't scar as much as older folks, but then there are folks that form what are called keloid scars, and you've probably heard of them. folks that form keloid scars actually have really, really, really active fibroblasts that lay down lots and lots of collagen. And the scars can actually be disfiguring. They can be so thick. And that's a genetic trait. And we find it in some groups versus others. And in some individuals, they actually go in and they try and remove, once the scar has done its thing, they actually try and remove the scar surgically to try and minimize it. But yeah, I imagine it's just our body's way of trying to protect us and to prevent invasion of pathogens, but also to restore continuity of a tissue as quickly as possible. But it's a good question. The spiny mouse, the African spiny mouse, I've been told that it can lose up to 60% of the skin off of its back by a predator coming down and grabbing it. And it can run away and repair and replace that skin without scarring. So the folks who are really interested in trying to figure out how to quiet scarring in us are busy comparing that mammalian system to other mammals in the laboratory and what genes and proteins are turned on and expressed during that process that are different from each other, why one mouse scars and the other one doesn't.
I imagine the spiny mouse doesn't regenerate like the axolotls do, but somehow at least it's moving in that trajectory.
Well, I think they can regenerate their ears. They're not
wow.
ear punctures. Rabbits can do this too, though. You can take a paper punch and you can make a punch in a rabbit's ear and it'll regenerate. And part of the reason we know that they can regenerate ear cuts is because sometimes they mark animals with clipping their ears in experimental studies, and they restored. Now, let's talk about digits and regeneration. So up until young adulthood, if you lose this much of your fingertip, so there's a portion of the nail bed still there, it'll regenerate, it'll grow back. And so in axolotls and during this regeneration, what happens is there's a wound epidermis that crawls over the surface of the stump, comes in from the side, and it seals it. Now in axolotls, if I amputate an axolotl, before I put my head on a pillow that day, the epithelia has crawled over the surface of the stump and sealed it. The wound epidermis seems to be really important because in an axolotl or fingertip, if I sew it up, no regeneration takes place, axolotl or fingertip. The wound epidermis crawls over the surface of the stump and it is able to send a message to the cells right in the stump to undergo de-differentiation, stop looking like muscle, skin dermis, look more like a fibroblast, and it forms a regenerating bud called a blastema. The blastema proliferates, divides, grows, and in time will restore that distal tip. Now, it might be a wee bit shorter, but for children, the fingertip comes back. For adults, they're much more likely to say, I've got to get back to work, sew up my finger, and I'm going back to work. And if I take a newborn mouse and I cut off its fingertip, and this experiment's been done, it regenerates very well. If I go one joint in me or the mouse, it can't regenerate. And so there are a number of labs in the country and in the world who are really working very hard to try and figure out how to determine why can this regenerate, but this can't. Because once we figure out the subtle differences in the wound healing process and the regenerative process, then we'll have the ability to regenerate distal tips for ourselves, but also more importantly, the joint in between. And that's a lot of complicated stuff. That's not just, growing out your bone tip to look the right length. This is lining up the ligaments and the tendons and the muscle attachments and all that other stuff. But people are really looking hard at trying to figure out how to get this to go. Now, an axolotls regenerate their arms, their legs, their tails, their gills, their lower jaw, portions of their heart. I can make an axolotl a paraplegic or a quadriplegic. I can cut its spinal cord, completely transect it, and it regenerates. Axolotls can take a pretty good hit to the forebrain. You can actually remove a portion of its forebrain and it regenerates. So what we'd really like to do is figure out how that happens at the molecular level. We know about the cells, and we know the cells that divide, that seal the pieces together. But what we don't understand is, why do the nerves just grow through that wound so beautifully? Whereas for us, we have these cells called astrocytes because they look like stars. They are very important for the blood-brain barrier in our nervous tissue. But the astrocytes are the ones that lay down scar tissue when our nervous tissue is damaged. And so when somebody damages their spinal cord or bruises it on the football field or wherever, the first thing they do is they take that individual and they make them very cold, because they're trying to slow down the inflammation response and the astrocytes and the scarring response to allow for the neural tissues to recover and hopefully maintain their connections. But if it's transected, it's game over. And that's what we're working really hard to figure out, is why can an axolotl regenerate its spinal cord and heal that wound and grow those cells through to restore function, and why we can't. Now, there were these really interesting experiments that were done with newborn rats, and they go in and they transect the spinal cord. They basically make the rat paraplegic, can't use its hind legs. But in some of their experiments, they made the cut, and at the same time, they grafted in a wedge of fetal. So a rat pup that was still growing inside its mom, they take it out, they take a wedge of its spinal cord, almost like a piece of carrot, and they put it in the cut space. So it's newborn rat cut, fetal rat wedge. And in a good percentage of those experiments, the fetal spinal cord tissue encouraged the neurons to grow through, and the rats were able to use their feet again. And so the embryonic environment and the molecules that are present in an embryo seem to be very different than what happens after you're born. We don't need to be grafting fetal tissue into spinal cords. We just need to figure out what the signals are, the molecules are, the matrix recognition that allows us to put this sort of magic glue in a spinal cord cut that hopefully will discourage scarring and encourage growth and healing. But we're not at that place yet, but we're working on it.
Yeah, I can imagine. So like you talked about cutting the skin, or that your skin kind of is directional. It grows to strength in one way. And then when it's healing, it probably is just trying to fill the space. It doesn't know which direction to go there.
Exactly.
And then that gets insanely more complex when you're talking about a limb or spinal cord or something like that. The organization of the cells, they need to have a bigger picture, basically, somehow.
It's really cool you said this, because I wasn't going to go there, but the experiment where they put the little wedge of fetal rat spinal cord in the cut, if they aligned the polarity, so anterior, posterior lined up, they got a better result than if they flipped it and we had anterior meeting posterior before it went to anterior. So the orientation of the little piece made a difference. So the cells can tell. They can tell anterior from posterior, right from left, grow this way, don't grow this way, walk this way. So it's really, and what we need to do as scientists is figure out, what are those subtle clues? And now with molecular biology, you can take a single cell and you can figure out what genes are on in that single cell. So experiments that we couldn't have imagined 15 years ago, we can now start to tweeze apart. and see very subtle differences in groups of cells. What's going to make a heart, what's going to make a liver, what's going to make an eye, what's going to make the forelimb, what's going to make the hind limb. And we can start to say, how are they similar and how are they unique? And how do we turn things on and turn things off? And so in development, we want to understand how they turn things on. We also want to know why things stop and don't overgrow, because that helps us to understand questions about cancer and why a cell population will grow unruly and not differentiate into the structures they normally would. There are these really cool experiments were done a long time ago with a group of cancer cells called teratocarcinoma cells. These cells are odd in that they divide and they differentiate into lots of weird stuff. You might have a ball of cancer cells that might have an eyeball or a tooth or hair growing one way. This is like your worst nightmare of a growth. So there was an experiment where they took teratocarcinoma cells from a black mouse. If they inject those black cells, those teratocarcinoma cells into a mouse, it'll make cancer. But they took those cells and they injected them into an early embryo of a mouse, into a place called the blastocele, the fluid-filled space in the middle of the early embryo. And they injected the teratocarcinoma cells from the black mouse into a white mouse embryo. And they implanted that embryo back into its mom, and a mouse was born. And the mouse was a mix of white cells and black cells, and it was perfectly normal. So cells that are in an adult environment will make cancer. In an embryo environment are re-specified, restrained to participate in normal development. And that's all a question of the signals, the clues, the touching, the rules and regs of an embryo saying, no, we're not going to make a teratocarcinoma cell. You're going to make one of the eyeballs. And they do. So you get a mouse that's got a black eye and a pink eye, patches of black on a normal white mouse background, and the embryonic environment retrained. That's why it's so important for us to study embryos, because the embryonic environment really holds the key to so many of these diseases and understanding them.
So I was listening to a podcast that he was talking about some mechanism that they do this in animals that can regenerate. And they had a way of turning off this communication, he called it. And basically, when you do that to an animal that can normally regenerate, like an axolotl, that it won't anymore. And also he said then axolotls basically don't get cancer. And he said if you do this same process of not letting the cells talk to each other, you can also give them cancer that way.
Interesting.
I think the more famous one maybe is he rearranges tadpole faces and then they grow back into.
Right. So, I mean, there's a lot of experiments that have been done where you can, so if I take a human embryo and I split it in two early enough, I can get identical twins. That's easy. We all know people who are identical twins. In a mouse situation, imagine taking an early mouse embryo and then another early mouse embryo and another early mouse embryo, and you sort of smush them all together. So we've got 6 different parents made 3 different embryos, and we smush them all together. So you'd think you get this monster with parts going every which way. If you do it early enough, you get one mouse. It's the normal size. It has cells from all 3 embryos. It has 6 parents. And because its immune system develops after it's developing, it recognizes everything as self. So with basically, you can make a mouse with 6 parents. For my PhD thesis work in limb regeneration, I developed an assay that let us look at differences in wrist and elbow and upper arm level blastema cells. So backing up, if you amputate an animal at the wrist, it only can make wrist and hand. That's all it can do. It develops autonomously, true to self. If you amputate it halfway through its radius and ulnar, it's going to make the distal end of the radius and ulnar and the hand carpals and digits. If you amputate it halfway through the humerus, half of the humerus, radius and ulnar, carpals and digits. So there's what we call positional information along the proximal distal axis of the limb and a memory of that position in the cells. We know it's a memory because if the blastema grows big enough, before it's differentiated and made anything, you can actually remove the blastema, you can stick it somewhere else, and it's still going to make a wrist and hand. It's called the distal rule of transformation. It only makes stuff that's distal, and it develops autonomously. It's a long story, but in a nutshell, what I did was I amputated animals at the wrist or the elbow or at the mid-upper arm. And the same time, I also amputated the animal at the mid-thigh level, mid-lower leg. So say I've got an animal that's regenerating a forelimb blastema from the wrist and an upper thigh level blastema from the hind limb. And I wanted to know what would happen if I grafted the wrist level blastema, which is going to make wrist and hand, on the dorsal surface of the hind leg. So once the blastemas had formed, I went in and I removed a piece of skin and epidermis from the wound, from the blastema of the hind limb on the dorsal surface, and I stuck the blastema, the wrist blastema, on top of it, like this. And as the hind limb grew out and regenerated, the wrist blastema rode along. And it made a hand on top of the ankle. So I got a foot and a hand. If I grafted an elbow level blastema on top of the mid-thigh, it actually displaced down to the knee, and the elbow joint and the knee joint were together, and I had basically two limbs down the leg. And if it was an upper arm level blastema, didn't displace at all, it just stayed stuck. This was the first time anybody had ascribed a cell recognition pattern, a cell recognition property to wrist, elbow, upper arm level blastemas. So it was a big fat hairy deal. What was even more exciting is at the same time I was doing these experiments, which was in the early 80s, a man looking at frog embryos and frog larvae, young frog larvae, and treating them with something called a teratogen, a monster maker, a chemical that would perturb their growth and development. And the monster maker he was using was vitamin A, retinoic acid. Now, vitamin A is very, very popular these days. It's important in skin care and therapy. Kids that have really bad acne may take retinoids to treat that acne. And if you're female and you're taking retinoids, they want you to be on birth control. So you cannot have a child while you're being exposed to retinoids. What Iqbal Niazi in India discovered is that vitamin A changed the positional pattern and the structures that would form from the wrist level. Say, let's give an example of wrist. And then Dave Stockham and a guy named Malcolm Maiden in England, we explored this extensively. Normally, if you amputate an animal at the wrist, it only makes wrist and hand, and axolotls have 4-digit hands, so I'll do this. If I amputate an animal at the wrist, and 4 days after amputation, I inject that animal with vitamin A, regeneration is delayed, and a funny looking blastema forms from this level. And from this level in a dose-dependent fashion, so if I use the maximum dose, I can get a scapula, a humerus, radius ulnar, carpals and digits. I get a big extended long limb from the wrist. And if I give a little less retinoic acid, I might get a duplicated radius and ulnar. Give a little less, maybe just extra carpals. It's as though vitamin A is walking the positional information of the wrist blastema all the way back up to the shoulder girdle. So then I repeated my assay, taking blastemas from the wrist or the elbow or the upper arm, all treated with vitamin A, grafting them onto the dorsal surface of the leg, and they don't displace. So vitamin A allowed us to predictably modify positional information, and my assay allowed folks to visualize and see that cell recognition, cell adhesivity, stickiness, and identity go hand in hand, and that forelimbs and hind limbs recognize each other because wrists displace to ankles and elbows displace to knees. So forelimbs and hind limbs play by the same rules, but we still haven't identified the molecules that say, you're a wrist, you're an elbow, you're an upper arm. But we have some good clues, but I think it's going to be so subtle. You know, this is the middle forearm, this is the wrist, this is the elbow, this is the upper arm, this is halfway down the upper arm. Is it going to be sort of a slow spectrum of different molecules up and down the limb? And it probably will be. But a lot of it has to do with cell interactions, cell touching, cell sorting out, cell choices.
Yeah, it seems in some sense insanely complex that, you know, the way that all the cells have to organize themselves, but then also kind of simple. You give these simple signals through just a dose of vitamin A and it can react in a predictable way. I guess it's kind of similar to me if I think about like hormone treatment that people are doing now. It's on the news, right, with the trans stuff and that, but people take hormones, and the way your body interacts with hormones is incredibly complex. It has all these different effects on it, but it's like, it's a super simple thing for you to do somebody. It's just like, if you want to make them morphologically a certain way, it's just this one thing, and it creates all these changes.
Timing has a lot to do with it too. And I'll give you an example. If you have a frog embryo and it's divided a bunch, but it hasn't really started to change or differentiate or do this process called gastrulation, which is when cells move from the outside to the inside. And so if you've got this young frog embryo and you take some cells from what will be the future belly of the frog and you stick it on the top, those cells can actually participate with their neighbors and actually differentiate into neuron, okay? Because they're in the right place at the right time early on. You wait another number of hours, like 8 hours, 10 hours, you take those same cells and you stick them right in that same spot. And they're like, huh, I'm not going to play with you. And they'll actually migrate all the way back through the embryo and find where they should be. And so cells have a developmental potency and a developmental fate. And early on, they may have more potential to do a variety of different things. But then as development proceeds, they become restricted, they become committed, they become determined to just make that one thing. What changes? Those are subtle things that we're looking at to try and figure out, like, what makes a cell determined to be nervous tissue and why 4 hours earlier it doesn't care what it does. And it's all because of cell interactions and the numbers of divisions and its neighbors.
Is that maybe at least part of why axolotls are so good at regenerating? Because I heard they're actually like in terms of their stages of maturity, they're basically juveniles, right?
They are. All right. So it's, and I'm glad you asked about this because we know a lot about it. So there's this axis of hormonal interactions in our brains, anterior pituitary, thyroid gland. So your hypothalamus talks to your anterior pituitary, which causes it to release something that talks to your thyroid gland, which causes your thyroid gland to release thyroid hormone. So axolotls have a defect in that pathway. They don't release thyroid hormone. They don't go through metamorphosis. In my lab, and a paper I published, gosh, back in 1998, but I wasn't the first to do it. I just did it very well. If you inject a small amount of thyroid hormone into an axolotl, it'll go through metamorphosis. If it's a wild type axo, it'll look just like a tiger salamander. And it resorbs its gills and its tail, it changes its pigment, it develops eyelids. it's the whole head morphology changes, the digestive tract changes, everything changes. So what I'm giving the animal is a little bit of thyroid hormone. That's the metamorphosis signal. Well, the anterior pituitary signal to the thyroid gland is called something called thyroid stimulating hormone. So if you give an axolotl thyroid stimulating hormone, the anterior pituitary signal, it'll talk to the thyroid gland and it'll metamorphose. So we go one more step up the stream, the hypothalamus. If I give an axolotl the hypothalamus signal, thyrotropin releasing hormone, nothing happens. Axolotls have gotten kind of stuck in the water. They can't go through metamorphosis. They can't leave the water. They keep their larval gills throughout life. And that also might be part of the reason why they heal so well, because they're sort of in this juvenile state. And folks have looked at the immune system of this animal, and they've seen that the immune system is also very, very important in allowing for regeneration to occur. And maybe that's why if they modify the immune system, they can get cancer. If they modify the immune system, they lose their ability to regenerate as well. I think our best hope in understanding axolotl limb regeneration is understanding healing, understanding joints, understanding how nerves can repair and grow and hook up. I think that's our best shot with the axos.
We can start wrapping it up here. I do still have a question about how evolution selected for this trait. Like, is there trade-offs that go with it, or why did they get this superpower? Because it is like a superpower. Like, literally, we have, superheroes that that use this as their superpower.
Well, there are a lot of animals that regenerate. I mean, hydra and planaria, simpler worms, starfish, of course, echinoderms can regenerate. Lizards self-amputate their tails and they regenerate long rods of cartilage. And I've been told that males that have longer rods than other males that have regenerated their tails are more lucky with the ladies. Maybe it's they were attacked and they lived to tell about it. I don't know. Deer have the fastest growing regenerating tissue on the planet. In fact, it's compared to cancer as one of the fastest growing tissues. So antler regeneration in some deer populations where males shed their antlers and then they grow these huge racks. So regeneration happens in a lot of organisms. Crawfish, lobsters, they can lose a claw, and when and they'll have no limb on that side, and then they go through a molt, and this teeny little bud will be there, and they go through another molt, and they'll have that limb back. And fiddler crabs that have this big claw and a little one, I've been told that in some species, if they lose their big claw, because the little one's here, right? And they lose the big claw. The next molt, the one that's further along now becomes the big claw. So that's another question of like, what's up with that, right? I mean, how does that happen? And so regeneration is a renewal and a repair process, but the axolotl is probably the master.
Do you see any like biological costs that come to this ability? Usually there's trade-offs to things like this.
Well, with trade-offs, I mean, axolotls don't live very long in the real world. They're endangered. They're trying to restore the wetlands. So they're native to 2 crater lakes. That's the only place they come from. But, and what do they give up? They certainly can't metamorphose. They can't leave the water. They can't explore other habitats. They're kind of stuck. So they're prey to their environment and the cleanliness and the health of that environment. I'll give you one quick example at the end, and then we'll tie this up with a bow. I had a student who was very interested in nerves and how nerves form. And so we used the green fluorescent protein animal, and we went into the arm and we removed a piece of the brachial nerve in the arm. And it's easy to find. It's like this white piece of spaghetti. The cells, the neural cells, are back up here in the spinal cord. So she was only taking the axonal processes from the nerves, but the supporting cells are called Schwann cells, and they myelinate the nerve and allow for nerve transduction to go more rapidly. So she grafted a piece of branchial nerve from a GFP animal into a white animal, into a limb of a white animal, and let it just sit there for about a week. And then she came back and she cut through the limb and through that piece of GFP nerve, and she let the limb regenerate. Well, not surprisingly, the Schwann cells, the support cells for the nerve, they were still alive and happy. And they proliferated like crazy. And as the limb regenerated, the Schwann cells, the green fluorescent protein Schwann cells, went along with the nerve and labeled all the nerves in the whole limb. So basically it was a completely white animal with no fluorescent cells, except for all of its nerves were fluorescent. And it was brilliant. It was beautiful. And if science can be fun, that's what you want to do. Because if it isn't fun, it shouldn't be done, right?